Hello everyone, I am Aishwarya Gurme from Department of Biotechnology, Rajarshi Shahu Mahavidyalya, and today in this video lecture we are going to study about HPLC. Okay, then HPLC, what is meant by HPLC? What is the long form of HPLC? HPLC means high performance liquid chromatography. Okay, so chromatography. Chromatography is an analytical technique used to separate a given mixture sample into its components, and this principle based on when a mixture and mobile phase are allowed to flow over stationary phase. Then basically chromatography system consists of two phases. First is mobile phase and next is stationary phase. Okay, then what is the mobile phase? Mobile phase is a liquid or gas that carries the sample or a mixture over through the stationary phase, resulting in the separation. Then stationary phase. Stationary phase is a solid or liquid supported on the solid which remains fixed in a place on which different solutes are absorbed at a different extent. Okay. There are different types of chromatography methods. First is paper chromatography, then liquid chromatography, then gas chromatography, and last is high performance liquid chromatography. Today we are going to know about high performance liquid chromatography. HPLC is a type of liquid chromatography which is characterized by using high pressure and small particle size to push a mobile phase through the solution or column of stationary phase, allowing the separation of the complex mixtures with a high resolution. Components of complex mixture are separated by column and detected by at the exit by detector that measures their amount, and output from the detector is called the liquid chromatogram. Okay, this is the structure of HPLC. This is mobile phase, then this is degassing unit, then this is solvent delivery pump, this is injector which is connected to the column. Next, this is detector. What is the role of mobile phase? This is the mobile phase. Mobile phase which contain the organic aqueous solvent that are used depending on the analysis. Okay. And this is the degassing unit. It is used to remove the dissolved air from the mobile phase. This is solvent delivery pump. Okay. It is used to deliver the mobile phase at a constant flow. This is injector which is attached to the pump and column. Which injector which introduce the standard solution or a real sample to the HPLC column. Okay, and this is the column. Column is used to separate each compound's content in the sample. Basic definition. What is the basic definition of chromatography? Chromatography is an analytical technique used to separate a given mixture sample into its components. Then this is the work of the column, and detector is attached to the column which used to detect the carrier compound from the column. Okay. Next, these are the instrumentation of HPLC. These are the instrumentation of HPLC. First is pump, then degasser, then injector, then column, and then detector. We will see one by one. First is pump. The development of HPLC led to the development of the pump system. The pump is positioned in the upper stream of the liquid chromatography. Pump is positioned in the upper stream of the liquid chromatography. This system used to generate the flow of eluent to the solvent to the reservoir system. This is a degasser. When the gas is present in the carrier, this is detected as a noise and causes the unstable baseline. So degasser is present. Degasser uses a special polymer membrane tubing to remove gas. This is injector. Injector is placed next to the pump in this way. Injector is present next to the pump. This is simplest method which is used for injecting the sample which introduced to the flow of the eluent. Now column. Column, the separation is performed inside the column. The recent columns are often prepared in stainless steel housing instead of using glass columns. Packing material generally used is silica or polymer gel as compared to calcium carbonate. We use the silica or polymer gel as a packing material instead of calcium carbonate. Example, recently we used the housing of stainless steel column in this way. And packing material, stainless steel, this stainless steel column. And packing material generally used in this housing is silica, made up of silica or polymer gel. Instead of, as compared to calcium and carbonate, we use the silica or polymer gel as a packing material in the column. Next is detector. Separation of analytes is performed inside the column. Detector is used to observe the obtained separation in the columns. Detector is attached to the column and used to observe the separation of the components. Next, these are the types of HPLC based on the separation mechanism. Okay. First is normal phase HPLC. Also we call NP HPLC. Second is reverse phase HPLC. Also we called RP HPLC. Then third is ion exchange HPLC. And fourth is size exclusion HPLC. Now we are going to know about normal phase HPLC. Normal phase HPLC is a chromatographic technique based on the separation by using the polar is a stationary phase and non-polar is mobile phase. Normal phase HPLC is a chromatographic technique in which separation occurs using polar as a stationary phase and non-polar as a mobile phase. Okay, polar as a stationary phase and non-polar mobile phase. Means example, this is the stationary phase. This is the polar stationary phase and non-polar which are entering is the mobile phase. And this is stationary phase, polar. Stationary phase is usually silica or alumina which contain polar functional groups, which contain the polar functional group in stationary phase. The mobile phase consists of non-polar solvent such as hexane, heptane, chloroform, sometimes mixed with small amount of polar modifiers. Okay. When the sample is injected, its component travel through the column with mobile phase. When we injecting the sample flows with the mobile phase in the column. Okay. The polar compounds are strongly attracted to the polar stationary phase. The polar compound present in the stationary phase are strongly attracted to the polar compound of the sample which we are going to inject. Okay. The polar compounds are strongly attracted to the polar stationary phase, so they move slowly. And retain longer. Okay. The polar compound present in the stationary phase attracted to the polar compound in the sample and they move slowly and retain the longer time. Okay. The non-polar compound interact weakly and carries faster, but non-polar compound which are present in the sample are move quickly and carries faster as compared to the polar compound, because in that the polar is the stationary phase. Non-polar compounds interact weakly and carries faster. Separation mainly occurs due to the absorption and polarity differences. Yes. Next, opposite to that, this is the reverse phase HPLC. In reverse phase HPLC is the most commonly used HPLC technique. This is the opposite of normal phase HPLC. In that there is non-polar stationary phase and polar mobile phase. Non-polar stationary phase like this, non-polar stationary phase, and polar which are entering in the column is the mobile phase. Polar which is entering in the column. Stationary phase is usually silica particle bonded with non-polar group such as C18, means carbon chain 18, and mobile phase consists of water mixed with the polar organic solvent like methanol. When the sample passes through the column, non-polar compound interacts strongly with the non-polar stationary phase. When the sample passes through the column, the non-polar compounds present in the sample interact strongly with the non-polar compound present in the stationary phase and retain for the longer. Like this, when the non-polar compounds enter in the column which interact strongly with the non-polar stationary phase and retain longer, while the polar compound carries faster. The polar compound which elute faster, because this is opposite to that. Okay, non-polar compound strongly interact, but polar compound elute faster. Separation occurs mainly due to the hydrophobic interaction. Reverse phase HPLC is widely used for the analysis of drugs, biomolecules and pharmaceutical products because it provides good resolution and compatibility with aqueous samples. Now, this is the ion exchange HPLC. Ion exchange HPLC is a chromatographic technique in which separation of compound based on their ionic charge. The stationary phase contain ion exchange resins. In that, cation exchange columns have a negatively charged group. In this column, ion exchange column, cation exchange columns have a negatively charged group. Cation exchange column have a negatively charged group and retain positively charged ion, which retain the positively charged ion from the mobile phase, which interact strongly with positively charged ion. And anion exchange column have a positively charged group and retain negatively charged ions. And this is anion column, anion exchange column. Now, this is ion exchange HPLC. Ion exchange HPLC is a chromatographic technique used for the separation based on the ionic charge. Okay. Stationary phase contain ion exchange resin. Stationary phase which contain the ion exchange resin. Cation exchange columns have a negatively charged group. Cation exchange column. This is cation exchange column. Have a negatively charged group and retain the positively charged ions. This is cation exchange group. When mobile phase entered in the cation exchange group. When the mobile phase enters in the cation exchange column, in cation exchange column have a negatively charged group. In cation exchange column have a negatively charged group which retain the positively charged ions from the mobile phase. And in ion exchange column have a positively charged group and it retain the negatively charged ions. Okay. Opposite to that, in anion exchange column have a positively charged group, but it retain the negatively charged. Ion. Okay. In anion exchange group. This is mobile phase. Okay. When in the cation exchange group have a negatively charged group, but it retain the positively charged ions from the mobile phase. Opposite to that, in anion exchange group have a positively charged ion, but it retain the negatively charged ion. Okay. Mobile phase is usually aqueous buffer. In that, in ion exchange HPLC, mobile phase is the aqueous buffer. When the sample passes through the column, ions with opposite charge to the stationary phase bind strongly. Ions with opposite charges group bind strongly. Okay, and retain longer, while weakly charged or neutral molecules elute faster. In cation exchange column, example, opposite to that, when negatively charged ions entered in the cation exchange group, which elute faster because it is same. Mobile phase is usually the aqueous buffer. When the sample passes through a column, ion with opposite charge to the stationary phase bind strongly and retain longer, while weakly charged or neutral molecules elute faster by changing the pH and increasing the salt concentration. This size exclusion chromatography. Size exclusion chromatography is also called the gel permeation chromatography and also called the gel filtration chromatography. This is a technique based on separation based on their molecular size. Okay. In that, stationary phase consists of porous gel particles, like this. Stationary phase consists of porous gel particle which is small in size, This is stationary phase. And mobile phase carries to the sample to the column. And mobile phase carries the sample through the column. The large molecule cannot enter the pores, but these pores are very small. That's why large molecules cannot enter the pores and move quickly through the column. But large molecules can't enter the pores and move quickly through the column. So they elute first. But the small molecules enters in the pores take a longer time. But small molecules which enters in the pores take a longer time. Small molecules enter to the pores take a longer time and elute later after the larger particles. There is no chemical interaction between the sample and stationary phase, and the separation depends only on the size. In this size exclusion HPLC. Size exclusion HPLC is used for protein analysis, polymer molecular weight determination, and desalting of biomolecules. Okay. These are the applications of HPLC. Application of HPLC in pharmaceutical industry for the quality control of drugs. Application of HPLC in clinical and medicinal use for analyzing the blood, urine, and plasma samples. Application of HPLC is very important in food and beverages analysis for detecting the additives, preservatives, and sweeteners. Application of HPLC are also used in environmental analysis for detecting the pollutants in water, soil, and air. Next, application of HPLC plays important role in biotechnology and biochemistry field, and its application also important in chemical and industrial application, and its application in forensic science for detecting the drugs, poisons, and toxins and analyzing the evidence samples. Okay, thank you.